All right, so here we go, part 2 as promised. A couple papers I want to show you guys first so that if you don't have them out in front of you, stop the video, go get them. First one, you guys have probably seen this chart somewhere along the line. This is your codon chart. You're going to need that. That's what we're going to be putting our notes on. We're going to be running through some practice examples on this one here. So it just says transcription, translation, practice worksheet. We're just going to do a few so that we get a hang of it. Eventually, you guys will be completing this one here. So it also has the chart on it, but then it has down the bottom some examples as well as some questions on the back. And somewhere in your possession, just because I want to look at the second chart that you guys are going to see. You guys will see a paper that looks like this. We're not going to complete it, but you guys will see a chart on it that I do want to go through. So if you have those 4 papers out with you, great, continue on with the video. If not, stop the video, go get them. I believe I attached most of them to the post that I put out this morning for you guys, but they should be in that packet of stuff that you guys picked up. So we're going to start with the codon chart itself. So this codon chart is going to kind of let you guys finish up your Construct Your DNA lab. And the big thing for you guys to realize is that this is a codon chart. You will be using your mRNA code. Remember, mRNA was made from DNA, so it's still DNA's code that we're utilizing, but now it's in a smaller form. It's a single-stranded molecule now, and now it's out in the cytoplasm where we're going to be making these proteins by stringing together amino acids. So the first thing that you're going to see is all of these around the outside of the circle here. All of these are my different amino acids all the way around. So you should have 20 different ones. Some things that you guys will see on here are things like stop. So stop is not an amino acid, but these are going to be my stop codons. Remember, that is going to be our third step of translation. When we hit one of these codons, that's where our protein's going to end. But the rest of them are your 20 different amino acids. Please be aware that you may see them in multiple places. So for instance, I might have, let's see, I think leucine is going to be here, and then it's also going to be, maybe that's not a good example, also up here. So it's not just found in one location along the chart. You may see them in multiple locations. Remember, we have 20 different amino acids, but I have 64 possible combinations that I can arrange this. So they're going to be coded for by more than one codon here. So the first thing we're going to do is we want to start in the middle. And we're going to start with these big letters. And here's the first letter of my codon. So we're going to say, oh, do we have a U, a C, an A, or a G? So this will be step number 1 that you guys will have here. We'll start here and we're going to work our way out. So if you're like, oh, well, my first codon starts with A, I'm going to close the rest of these off, which means that I know I'm only going to be working within this particular section. First codon. My second codon is coded for by that second ring that you guys see here. And we're basically just working our way out. So let's say I'm going to look for AA. I know I'm in this little box here. Here's my first A. Here's my second A. Now I know that I'm only working within this area here. So then I want to look for my third codon. And my third codon is going to be the outermost ring. So I'm starting inward and I'm working out to my amino acids. And that's what you guys are going to see. So first base pair, let's say this would be my second within the codon, and this would be the third within the codon. So if I'm going like this and I say, oh, let's see what GAG represents, that's going to give you your amino acid on the outside here. If I said AAA, that's going to give me lysine. What we want to look at is my stop codon. So we did say that there are 3 stop codons here. So if we're looking at them, we can kind of work backwards and go in this direction. So I know my first stop codon is UAA. I know that I also have UAG, represents stop. And my other one is down here. So we're still within the U's, but now G is that second base pair and A is the third base pair. So I have UGA as that third one. So those 3 represent stop codons. That's that last step, termination of translation. It ends the protein. Remember, there are no amino acids that are going to code for this. It just says stop. We also have a start codon. My start codon, if you remember that messenger RNA strand as it goes through the ribosome, that ribosome is not going to start making the particular protein until it reaches this 3-letter base pair. And my start codon here happens to be what is known, coded for by what is known as methionine. So it's going to be I'm sorry, AUG is going to be my start codon. As soon as that ribosome reads that particular codon, that's when it's going to start stringing together those particular amino acids. So AUG codes for the amino acid methionine. It is the only codon that's going to code for methionine. So down the bottom, I put some little practice ones for you guys. And I gave you the messenger RNA strand, so I can say, oh, this is my 5' end, this is my 3' end, because I know messenger RNA is going to get read 5' to 3'. So this end's going to enter the ribosome. He's going to start reading those particular codons, call in the correct transfer RNA who's holding his amino acid. So if I'm given that messenger RNA code, I can kind of figure out anything along the way. Maybe I want to go and do DNA first. So if I'm doing DNA first, I know that A is going to go with T. I do have thymines here. So I have T, T, T. If I have U, U, U as my codon, now I'm going to go A, C, G, A. ACU should be T, G, A. Remember, uracil is going to code with adenine. I still have adenine in DNA. CGC codon is going to represent G, C, G. So in the blue, that top strand there, those are my triplets. These are my codons. This is what I'm reading off of that codon chart above. I can then go through and transcribe it because I know that my anticodons in transfer RNA are going to be complementary to my messenger RNA. They are matching up at the ribosome there. So if I say, oh, I have adenine, 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 or AAA in messenger RNA, I'm going to have U, 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 because in transfer RNA, my thymine gets replaced by uracil. Over here, I have A. U goes with A, U goes with A, U goes with A. So if you guys finish that up, that's what you're going to see. You're going to notice that your DNA and your transfer RNA are going to look pretty similar to each other. And the reason is they're both complementary or matching to that messenger RNA. The only time they're different is that DNA's got thymine, tRNA or your anticodons have uracil. That's the only place they're going to be different. Other than that, they are identical because they are both complementary to your messenger RNA. The last column we have there, now we have to find our amino acids. I do not want to do this. I don't want to go, oh, well, transfer RNA is bringing in the amino acid. It's still the code from messenger RNA that came from DNA. So I have to read my messenger RNA codons. It's why it's called a codon chart. So my first one is AAA. I go up, first base pair A. It gives me my amino acid lysine. I'm just going to put the first couple letters there. I know that these amino acids are going to be strung together with a peptide bond. The next one, I'm going to go U, U, U. So here we are up here, first base pair, second base pair, third base pair, I have phenylalanine. So I have a PHEN, another peptide bond. And I have GCU. Here's my G, there's my C, there's my U. I have alanine, another peptide bond. We're going to keep going. ACU. And my last one, CGC. So here's my C, G, C, and we have arginine. This would keep going until it read a stop codon. At that point, it would break off, and we're going to kind of talk about what happens with that amino acid tomorrow's notes. There's a second chart that I want you guys to be able to look at. So on this sheet here, you guys will see a little box one. I'm going to use a bigger version, but you guys have the box one there. It gives you the same information that you have. So if we're looking at the box one that we have here, my first base pair is going to be down this side. My second base pair is going to be across the top. And I'm going to kind of connect them all here in a moment. Where do they all meet up? And my third base pair is down the far side. It gives you the same exact information. I want you guys to be familiar with it. I like the wheel one better, but I know on the Khan Academy, as well as some state tests and some other books, are going to be using this particular chart. So let's look at AAA, which was the first example on the codon chart. I'm going to look for my first base pair, A, second base pair, and I know that now I'm going to be within this box. My third base pair, also an A. I'm going with lysine. So basically, where are they all connecting? If I'm looking at UUU, which was our second codon on the other chart, U, match it together here, and my third one, all the way across, I have phenylalanine. If I was looking for my stop codons along the way, I have one here, here, and here. So I can go, oh, it's U-A-G, U-A-A, and my third one is going to be U-G-A. So you can kind of work backwards on it. It's going to give you the same information, whichever one that you guys like better. So if we're looking at the practice sheet, just so we can go through the little one, and this is completely for you if you guys need more practice, you want to send it back to me to check on, perfect. But here's my DNA. I find it easier if I go through and just kind of separate them because I know every 3 base pairs is what I'm going to be looking at. And they kind of strung this one all together on you guys, but you kind of want to go through and look at every 3 base pairs on it. So I know that my DNA can be turned into messenger RNA. Every 3 base pairs is going to be a codon. And then I can transfer that, and that's kind of what they did the next line. I said, oh, okay, well, here's my codons. And they kind of put them through within that. And then they gave you guys anticodons as well. So you need to utilize these codons in order to calculate out your amino acids. And that's what you guys see here. You want to use this particular line in order to do that. The worksheet that I want you guys to do, and that will be due on Wednesday, I'll put the assignment in, looks like this. So it'll be on Google Classroom. You guys will turn it in through Google Classroom. You're utilizing this. I would also, when you get to the back side, have this one out next to you so you guys don't have to keep flipping back and forth with it. But it's just giving you guys a chart. So if you know one bit of information here, you guys should be able to figure out the rest of them. So if you're given messenger RNA, you guys can go and figure out your DNA. You guys can go and figure out your tRNA. You guys can go and figure out your amino acid. So for instance, we'll do the first one together. AAA, if I make it into DNA, I would say that A goes with T, A goes with T, G goes with C. If I put this into tRNA, my anticodon, I know that A goes with U, U, C. Now I want to look for my amino acid. Use your messenger RNA, not this column. You guys will see that example or that answer on your test. I'll make sure both of them are there. Know which one you have to use. So I'm looking up A, G. I know that my amino acid is lysine. Okay, so you guys are completing the rest of this chart utilizing this codon wheel as well as what you have on the back there. Okay, so that will be due on Wednesday. Let me know if you have any questions. Post them on Google Classroom under the comment section so that others within your class are able to see them. Check out the con videos that you guys have there. There's a lot of practice tests at the end of that particular section. And remember, we'll be testing on Friday, so the rest of it's going to kind of be reviewed. Tomorrow, you guys will see the rest of the diagram notes, what happens once we make the protein, and we'll go through all the things before we start that review. Let me know if you have questions. Thanks, guys.